There's another person that I would like to thank today who is here from Google, and it is Javier Gonzalez. And he is an amazing partner. Many of you in the room know him and his counterpart, Rebecca Prosen, who wasn't able to join us today, but thank you, Javier, for all that you do for our communities. So our second topic of discussion today is the life science industry. And I am really pleased to bring up our next speaker, who I have known, gosh, probably 15, almost 20 years. We started when we were very young. Let's keep that in mind. And I think many of you in the room have interacted with Carla Boragno at some point in her different roles. She is a former senior vice president, head of global engineering and facilities, pharma technical operations, Roche, Genentech. She is born and raised in San Mateo County. She has been a tremendous resource to our life science organizations, Biocom, Michelle Nemitz we have in the room, California Life Sciences. And so I want to bring up Carla to talk about the life sciences industry.
Great, thank you. Great, thanks so much, Roseanne. And it's a real treat to be here. And so as Roseanne mentioned, I just want to start by talking about my journey in San Mateo County. So except for college and a 2-year overseas assignment in Southeast Asia, I've lived my entire life in San Mateo County. So I was raised in Millbrae, went to Capuchino High School. My dad, is there another Mustang back there? My dad worked at the Bethlehem Steel plant in South San Francisco for 30 years and ended up running that plant until its closure. Shortly thereafter, I graduate college and I get my first job at some little small biotech company that I hadn't heard of and most people hadn't heard of at the time, and it was Genentech. So in 1986, I started driving to work the same way my dad did, right at the time when South San Francisco was transitioning from steel mills and paint factories to the birthplace of biotechnology. And I finished my career there just last year when I decided to retire. Happened to meet a guy about 35 years ago who grew up in Foster City. In fact, his parents were one of the first 25 families to move into Foster City in the early 60s, back when everyone thought that the first earthquake, right, Foster City was going to go into the bay. And San Carlos has been our home for the last 30 years. So again, just couldn't be more thrilled to be here with a lot of fellow county folks. And the other thing that I want to say is that I would never build anything, provide any advice, or do anything that would put my community, the communities where I work or worked, or my family at risk. So what I want to do today is have a conversation about that. Okay, so we're going to be talking about sort of the framework for biosafety. I want to give you a little bit of a background about what life science is, talk about the impact to the state and to the region, talk a little bit about biosafety. This is something that we could talk about for days, and we can go really deep and get really technical, which I promise I won't do. One, because I'm not a scientist, and two, we just don't have enough time. But I also want to just provide you with a little bit of the oversight and what goes on inside laboratories, and then we'll just close and have some questions. So what is life science, right? It's a term that's used a lot, and it covers many different aspects. So it could be biotechnology, it could be traditional pharmaceuticals. Pharmaceuticals are making medicines from chemicals rather than from biological agents. Medical devices, genomics, digital healthcare. For today's discussion, we're going to really be talking about biotech and the use of and biosafety in these labs. Okay, so let's talk about this impact to California. I'm going to let you read the numbers. They're impressive, okay? And this is the kind of impact that the life science and biotech has really had in the state of California. What I want to note on this slide is that when we think about the jobs in the broader life science sectors, not all of them require PhDs or bachelor's degrees. And that's actually something that oftentimes people think, or even high school kids think, well, gosh, if I want to go into biotech, do I need to study science because I really don't like biology, but there are so many different jobs at all levels. And I think that's super important as we think about this industry and this growing industry in the state. Now, when we get to San Mateo County, so the life science industry is the economic backbone here in San Mateo County. So think about this, $14 billion in impact to the county. And that's directly, from the industry, but also indirectly, right? Through vendors and suppliers and things of that sort. And I think what's also important here, and Roseanne mentioned this, is not only is there an economic impact, but think about the medicines and the treatments that are coming out of this industry. I have 2 people in my family that actually have benefited directly from products from Genentech, right? And there's so many different companies, right? Look at that, 800 companies just here in San Mateo County. And so this is an industry that is really doing something for good. And I think that's important to note. The other thing is that when Genentech started in South San Francisco, the first little office that Bob Swanson and Herboya rented was in this industrial park because the rents were really cheap. And there's some empty warehouse buildings. But more importantly, they wanted to be close to the major research universities. So UCSF, Stanford, Cal. By the way, that's where I went to college. So I still can't wear red if there's any Stanford people. There are some Stanford folks in the room. I'm brainwashed. Exactly. But with Genentech sort of setting roots in South San Francisco, what happened is it started attracting other major players. So today in San Mateo County, not only is there Genentech, we've got Gilead in Foster City, AstraZeneca, Merck have both set up but satellites in South San Francisco. And it's just really, I think, pretty powerful about where this industry is going. And it's a relatively benign industry when we start talking a little bit more about hazards in the labs. Okay, this is an eye chart, but my point is that life science and specifically biotech is a heavy regulated industry. In fact, I'd probably say that it's one of the most regulated industries. I know I did not get all of the logos up here, but I just wanted to give you a taste of the different agencies and oversight. The one that I do want to start with is the FDA. Okay, everything that happens at a biotech company has to be with the patient in mind, right? And because of the FDA, we can actually get medicines, go to the drugstore, get an Advil, knowing that it's going to be safe. And the reason why that's so important is it sets the framework for the safety mindset and the compliance mindset across biotech companies. And not just on the manufacturing side, but in the research labs, in the clinical trials, everything's grounded in that safety first for patients, always. And the FDA does not regulate for hazardous material. They don't regulate for lab safety or water or things of that sort. But because of that foundation of safety first for the patient, there's a culture of safety in these companies. And then you'll get all of the other, you've got the California Code of Regulations, you've got, the county agencies, local fire, but it's heavily, heavily regulated. And I did even put like NIH up there and CDC. So let's now get a little bit into the lab. So in a research and development lab, there's 4 kinds of hazards. Okay, there's chemicals, so work with chemicals. Not a lot of really exotic ones, but there are chemicals there. Radiation and lasers, small animals, and biological hazards. Okay, so again, we're going to be talking about the biosafety today, but just with any of these hazards present risks. And so there's mitigation measures that are used to manage these risks. So chemicals are stored in flammable cabinets, and those are things that are that the fire marshals sort of oversee. Okay, so let's talk about biosafety. So biosafety consists of practices in the lab, safety equipment, often called personal protective equipment, PPE, and specially designed laboratories to ensure that the worker is safe. But these protections also ensure that the environment in the community and people down the hall and maybe people, on the campus of the company are safe as well. But keep in mind, biosafety requires 3 things. Again, it's procedures and techniques, equipment, and then special design, the engineering design of the facility. Biosafety is governed at all levels. So you've got, local, state, and federal, California Code of Regulations. If you're working with biological agents, there's a whole list of what needs to be done, right? You have to have a biosafety officer. You have to have an emergency plan. All of these things, they're in the California Code of Regulations. Okay, and it's multiple agencies. There's not any one agency that oversees all aspects. So for example, medical waste is handled by the county. Now there are biosafety levels, okay, that talk about what kind of containment is needed. Okay, and this is where we start getting a little technical here, so I'm gonna try and keep it simple. But there are different biological agents and procedures, and based on what the biological agent is, will determine the risk level. And then based on what you're doing with that agent will determine what level of containment you need to have. And I've got pictures from the CDC on all of these things, so you don't have to remember all this. But by procedures, what I mean by that is the work that I'm doing with this agent, does it produce aerosols? Am I using needles? Am I handling small animals? Or are there high volumes and high concentrations? Okay, so again, there are some agents that present very, very low risk. There are some agents that have much higher risk, and then it depends on how you're handling that. So I'll give you an example. SARS-CoV-2 is considered, right, we all know that from COVID, is considered a risk group 3. But if a technician is working with a blood sample to do a testing, that would only require a certain level of containment versus if they're doing core research with live virus and handling a small animal. That would require a different set of containment. Okay, so again, it's the agent and it's what the technician or lab worker is doing with that agent. Okay, so now we'll get into some pictures, okay? So BSL1, biosafety level 1, is really the lowest level. And these are very basic labs that you might see at a high school. Although I'm guessing that there might be some high schools in the county that actually have more sophisticated labs, which I'll talk about in a little second. But teaching classrooms, maybe at some, healthcare offices, but it's very basic. So you're going to have a worker, they're going to have a lab coat on, they'll probably have safety goggles, you might have an autoclave, there'll be some signage in a sink, but very, very basic and not anything too sophisticated. Now, when you get into biosafety level 2, okay, this is now where you might be working with a biological agent, but it might be a biological agent that is very common, there's vaccines for it, et cetera. Now here in this lab, what you're gonna have is you're gonna have something called a biosafety cabinet. That's supposed to be, it looks sort of like a fume hood, but a fume hood takes away chemical vapors, where a biosafety cabinet keeps the worker protected from the biological agent or tissue culture sometimes. And you'll see these in diagnostic labs. You're going to see these in research labs. Almost every biotech lab has one of these. And this hood, this biosafety cabinet, also has a HEPA filter in it. So all the air that comes into the hood is filtered, and all the air leaving the hood is filtered, and it captures viruses and these biological agents, whether it's any sort of biological organism is captured. So the air that goes to the outside has got nothing in it. Okay, they don't deal with chemicals in these hoods. It's not a chemical fume hood, it's a biosafety cabinet, okay? And when you think about lab design, there's these levels of containment, okay? So primary containment, like a biosafety cabinet, is really to protect, the worker and the scientist or researcher that's most closely dealing with this agent. When you start getting into the additional levels of containment, whether it's secondary or tertiary containment, that's when you start looking at protecting the neighboring environments and the outside space, the air, the groundwater, et cetera. But again, biosafety labs, very, very common type of laboratory. Now let's get to biosafety level 3. This is, I think, what starts getting people a little bit nervous, because now in a biosafety level 3, you might be dealing with an agent that is a higher risk category and one where you may or may not have a treatment or a vaccine for, okay? So here you're gonna start seeing a lot more engineering controls to make sure that this lab is not only safe for the workers in the lab, but it's safe for other occupants in the building and safe for the community. So some of the unique features in these labs are the airlock, which is, oh wait, I didn't advance, sorry about that. It's like the airlock, okay? So this airlock allows for room pressurization. So if you imagine this is the lab, the hallway outside would be a higher pressure so that anything happening in the lab doesn't go to the outside, okay? And there's control doors and there's access. So only workers that have been trained in these labs can actually get into the lab. Okay, you've got your biosafety cabinet, but the finishes on these labs are all impermeable because these labs might need to be disinfected if you're changing agent or if there happens to be, you know, a small spill. So they have to be completely 100% sealed so that you can disinfect the lab. Okay, there are no floor drains. All of the pipe penetrations through the walls are sealed. Okay, HEPA filter air not only at the biosafety cabinet, but the ducts that take that air that has nothing in it, because there's a HEPA filter. When they leave the building, there's another set of filters, either on the roof or wherever these exhaust fans happen to be. And then anything coming from a sink would be contained in a separate tank, so nothing goes down the drain. And all of the work with these agents are still done in the hood, okay? The other thing that happens is that the researcher actually has additional level of PPE. So what you'll see here is probably a full sort of bunny suit and then a respirator just for the face. So it's not one of those blow up sort of suits, but it's just to make sure that the researcher has got fresh air all the time going into their sort of immediate breathing space. Now, these labs are rare. You're not going to have a lot of these labs. Where you'll see these labs is at probably every research university. So we know Stanford has a BSL-3 lab, UCSF, UC Berkeley, UC Davis, UC Irvine, right? So these major research institutes, the Gladstone Institute in San Francisco has one of these labs. And you might see them at some companies, some biotech companies, if that company is working on a vaccine for COVID, if that company is working on something, you know, more unique, some sort of infectious disease. But they're very, very, very rare, okay? And they're only used when absolutely needed. In fact, what the scientists attempt to do is design their experiments so that most of the work can be done in a BSL-2 lab, because these labs are expensive. They're expensive to build, they're expensive to operate, and they require a lot of inspections. They require a lot of third-party oversight of commissioning to make sure the engineering systems are all working. And they're tested and commissioned. And this is where I'll geek out on the engineering thing, so I won't go to there. But you want to really minimize the amount of work that's done in these labs, and you only do it when you absolutely need to do it. But again, there really just aren't that many around. I think there might, oh gosh, I don't even remember the number, but in the state of California, you just don't have a lot of these. Okay, so now we get to BSL-4 labs. And these are the labs that are dealing with the most serious agents. So when you think about hemorrhagic fever, Ebola, this is what goes on in the BSL-4 labs. These labs you will find probably only like at government-run laboratories or some very sophisticated university research centers. Okay, we don't have any BSL-4 labs in the state of California. Okay, they're even more rare than BSL-3 labs, but they're needed to actually start thinking about when, you know, the study of, you know, infectious diseases. Now, what's different about a BSL-4 lab from a BSL-3 is primarily the PPE. So this is when the researchers are in a bunny suit that's got positive air. So they sort of look like those Michelin men because there's air in there, so that if there's a little hole in the suit, right, there's pressure air, you know, going out. Also, you're required to take a shower leaving the facility. That's that little number 10 up there. And I think everything else is pretty much the same, but it's like a BSL-3 lab, it's airtight, but now the person working in there is attached to a hose that's filtering. I had the opportunity to tour one of these facilities earlier this year in April in Boston. So Boston University, it's where our daughter goes to school, has a lab. It's called the National Emerging Infectious Disease Laboratory. And they actually have a BSL-3 labs and BSL-4 in this facility. It's actually in the heart of South Boston as part of their medical school. And they're doing incredible research. And it's this kind of research that is going to actually help thwart the next pandemic, because we probably will have another, we don't know when. But this is the kind of research that is so vitally important that goes on in these labs to make sure. But these, again, are so incredibly rare. But the point in showing you these slides is that there's these level of containment to make sure things don't get out into the environment. So again, primary, secondary, tertiary containment. Okay, so now let's talk about the CDC and the NIH. The CDC has their primary, so one of their primary many responsibilities is to actually think about biosafety in these labs. So there's a document, it's a very, very long document. It's called the BMBL, so Biosafety in Microbiological and Biomedical Laboratories. And this is the cornerstone of all work involving biohazards. And this is where, this document talks about the required levels of containment in those levels that I just showed you. They talk and define the risk groups. They talk about what's needed. And they also talk about how the facility should be designed. They've got actually a checklist, and it's 6 pages long with very small print of all the things that need to be done in order for people to be safe, how agents are transported, whether it's a low risk or a high risk, what the appropriate techniques should be in the lab, how you account for inventory, et cetera. So it's very, very detailed. It's the cornerstone. It's written by over 200 scientists, and then there's new editions that come out. And so again, it's very, very detailed. Okay. And it's really, I think, something that assures that this work can be handled safely. And then the last thing I want to sort of share with you is something called the Federal Select Agent Program. So this is also run in partnership by the CDC, but also with the Animal Division as well. And there are, let's see, there's a list of, it might be about 50 agents that are the most serious, okay? Almost all, I should say, all the BSL-4 agents, or that you would work on in BSL-4, would be covered under this, and the majority of agents that would require BSL-3 facilities. So this is a program that really ensures compliance and provides the oversight for these agents, okay? And so there's a lot of data out there, but you'll see the majority of these agents are done, at the federal government level, and academic level, et cetera. And they actually have to understand what the nature of the research is. They will do security background checks on the principal investigators, and they will inspect the facility before they will issue a permit for these labs to operate, okay? And why that's important is that if one of these scientists wants to get very, very small volumes of these agents to conduct their research, they need to have a permit number in order to be able to get a hold of this material. Okay, and a lot of this is to ensure that things don't get into the hands of bioterrorism or things of that sort. In fact, I was telling somebody earlier that the new woman who runs the lab at Boston University, the Needle, she joined in January. Nancy was the former chief of the biodefense unit at the CDC. So again, it's highly regulated. There's inspections. These guys do drop-in inspections as well. They look at inventory materials, et cetera. And it's to really cover those things that are on that list. So again, SARS-CoV-2 falls on this list. Anthrax is on that list, and some other sort of nastier agents. But typically, you're not going to see this in your everyday biotech company just because it's so rare. But just another example of the kind of oversight that happens. So what I want to close with is the industry is highly regulated. Okay, I think I've talked about that over and over again. There's multiple agencies, okay? And all the work that's done at biotech companies involves risk. But there are measures and mitigation measures that you put in place to mitigate that risk. Okay, and it's part of the accepted practice. It's how scientists are trained. It's how these companies run. And you want to have those so that you can ensure you've got not only safety for the people working in the labs, but also for the employees and the community and the environment. Okay, and like I started when I was talking about the FDA, life science companies are highly motivated to ensure safe operations. And so there's a lot of times where people talk about safe science is good science. Because to not have a safe environment or to not do something that might have an issue, with the FDA, it costs a lot of money. And again, with, young companies and early stage companies, they're requiring money from venture capitalists. And if something goes wrong, shuts them down, it's just not good. And it's not good for the people working in the labs, okay? So I think that's it. The thing to remember is safe science is good science. And lastly, I think it's super important that flexibility is provided for life science companies, okay? Because in order to grow the business, in order for them to continue the work that they need to do, they've got to be in communities where there's flexibility, where they can actually continue to develop, life-saving medicines. And so that's something as well that I'd like to leave you with. So thank you, and I think now we're going to have some Q&A. Do you have a set chair you want?
No, wherever you'd like to sit. I'll come over here. So I want to start with acknowledging something that Carla said at the get-go. She was born and raised in San Mateo County. She left for a very short time to go to that school across the bay. Go Bears. Come on, let's have a go Bears. Let's have a go whatever other university you're from. I know I have a lot of Cardinal fans in the audience. But what Carla said was she would never do anything in her role at Genentech Roche for 30 plus years that would harm her community, her family, the workers, and the industry. Now, let's think Carla's one individual who works for a life science company. How many thousands of people work in this industry in San Mateo County today? I look at our council members from Foster City, from San Carlos, from Belmont, from Redwood City, and I think about South San Francisco. I think about those communities. These are our friends and neighbors, folks. These are the scientists. These are the professionals within the lab and outside of the lab. These are the parents of children in our schools. They all think like Carla. I would be hard-pressed that you would find one of them that would not utter what she said at the start of this presentation. How many of you all, I'm going to ask you the same question, learned something in Carla's presentation that you did not know when you walked into the room today? Okay, again, pretty much every hand in the room is up. So I have a few questions for Carla, and I'm just thinking about where we are in our world today and the just onslaught of information and things that are happening. And one of the challenges I think policymakers face is the questions and concerns regarding BSL-3 and why would they consider allowing them in our communities in San Mateo County when there are so few to begin with? If you could expand on that. You touched on it a little bit, but it'd be great.
That's a great question. So I have a phrase. It goes like this, if I can get my words right, is that scarcity of research should not equate to scarcity of resources. Okay, so what I mean by that is work in BSL-3 labs has to happen, right? For all of us that have received the COVID vaccine, it required a BSL-3 lab, okay? Now, just because there aren't a lot of need for these labs doesn't mean that we shouldn't have them. Okay, and it actually sort of reminds me of affordable housing, right? Everyone's like, yep, we're all for housing, but just certain kinds of housing, right? And let somebody else, right? It's the NIMBYism. Let somebody else worry about, the affordable housing. And I think that's what I think of. And I'll give you an example. So has anybody heard of Vir Biotechnology in San Francisco? Yeah, a few. Okay, so Vir, they are, I would say, they're not early stage. They're well funded. They do some pretty amazing work. They don't have anything really commercialized yet. But they study virology, viruses, et cetera. And they're based in San Francisco and Mission Bay. And when the pandemic hit, they were approached and asked, could they actually work with the CDC and NIH on work with COVID? Yes, we can. We need to build a BSL-3 lab. And because San Francisco allows this kind of work, they were able to quickly build their BSL-3 lab. And I think now they might have two and maybe building another one, so that they could do the important work that was needed to be able to understand what was going on. So that's an example of where a company may not need one of these labs today, but right, we know that our company portfolios are fluid and research changes. They might need one in the future. And there's unintended consequences, I think, sometimes of decisions that are made at the policy level. And I'll give you another sort of example, in one of my jobs at Genentech, I used to help make the recommendations, depending on the size of the project. Sometimes I wasn't the decision maker, but where we would make our real estate decisions. In other words, what local cities would we go to if we were to build another manufacturing plant, if we needed another sort of office or something. And what was always important for me when we were looking and doing our search was, does the community have a skilled workforce with the right skills that we need? Is the city keeping infrastructure growth at a pace that we might need for us to grow as well? So infrastructure, whether it's water, sewer, roads. Is the local city going to be innovative and partner with us? Are they going to be a good partner with us so that when issues come up, we can solve things together and work together? And the last thing is, are they going to make science-based decisions? And so that's what I used to look for. So now let's sort of have a hypothetical situation. I'm an early stage biotech company, don't really have enough capital yet to build my own 1 million square foot research center. So I'm going to actually decide to rent some labs from a developer. And we see all the development that's going on up and down the peninsula. Now, I might not ever need a BSL-3 lab because my company might be focused on oncology or some, eye disease. But am I going to, make a huge investment in a 5 or 10 year lease, which for an early stage company is probably the single largest investment that they're going to be making, right? In a city that maybe is not going to be good to work with. I'd go somewhere else because I don't know what I'm going to need in 4 or 5 years' time. So I think that there's some unintended consequences of decisions that are made. And so, again, I'll go back to just because there aren't a lot, we need to make sure that when it is needed, because it's going to be needed for something that's super important that might impact all of us, we want to make sure that we've got the facilities necessary.
Thank you for that. And I think one of the other questions that faces policymakers is, how do they address the real fears that community members bring to them? I mean, people are scared. And so I'd love for you to comment on that.
Yeah. Well, first of all, to the elected officials in the room and those of you that are representing elected officials, thank you for your service. You guys have hard jobs, and I really respect the things that you have to balance. So I want to start with that. When there's fear like that, I think my first guidance would be to listen. And I don't mean just like, yeah, come on, really, like, this one's, writing me all these emails, all these, but really, really listen. Okay, because that fear is coming from somewhere. And most likely the fear could be something that they've read, something that they hear about, hopefully not something they see on the kids' channel of YouTube, but that fear is real. And I think it's important for elected officials to sit and listen and to engage, because it's about building trust, okay? And the first step in building trust is listening and trying to understand. And I know that when I was dealing with my teenage daughter and she was upset about something, if I'm trying to push logic on her, it wasn't going to work, right? So you've got to really find a way to diffuse that fear. And you may not ever be able to convince somebody, but to listen, make them part of the process. And there's multiple ways, right? I mean, you guys probably know better than I do, but it could be discussions, community engagement. In fact, I'm going to go back to Boston because it's such an interesting example. Is the Needle has a community liaison program where they actually have 2 community members that meet on a regular basis with leadership at the Needle. And the Needle they're transparent about everything. If they have any sort of incident, it could be a needle prick or something, they listen, they put it on their public website. And so the other, I think, important role that elected officials can play is how are you going to partner with the companies in your communities to establish that kind of transparency and interaction with the community, because that's what will build trust. Okay, so that's, I think, the first and most important thing. And then at the end of the day, you're going to have to make a decision, right? And you have to look at, this is where now the facts and the logic come in. And you have to make those decisions based on, okay, there's agencies that oversee this. There's safety precautions that are in place, right? You know, you have to look at the risk and you've got to balance those decisions. But dealing with that fear always goes back to listening and building trust. And sometimes it's making the decision to do what you feel is going to be best for your community. But I'm not going to sit here and tell you that there's zero risk. There's nothing that's zero risk, right? I mean, those of you that have airports in your communities, there's risk of having an airport, right? There's risk when we put a stop sign up or choose not to put a stop sign up. It's just that, are there the right measures in place to mitigate those risks? Is that, yeah. Yes.
And could you remind everyone the Needle? That's the Boston.
Yeah, it's the National Emerging Infectious Disease Laboratory. It's really impressive. It's and you know, it's sort of fun now because now that I've had a chance to meet Nancy, and we've sort of developed a great relationship. But they do very impressive work, and is really around looking at, you know, what's going on globally with infectious diseases, and how the work that they're doing is hopefully going to be able to really understand, you know, how these diseases mutate, how they jump from species, and we need that kind of research going on. Now, let me preface it by the research that's going on there at the Needle or at UC Berkeley is not what goes on in a biotech company. It's very, very different. And it's, you know, at these university centers, it's basic research. It's trying to understand those big questions. So they're looking, and that's why they have the needs oftentimes for these very sophisticated laboratories. You know, a biotech company says, I think I have an idea or a molecule that might work against this particular virus or this particular cancer. And so they're going to be doing very specific experiments when they have to go into a BSL-3 lab to say, will this molecule actually do what I think it's going to do? It's not this sort of basic research. And I think that scares people sometimes too. They read things, and, you know, we all know that we still, I don't think, are ever going to know, you know, what started the pandemic, right? Did it come from the lab? Did it come from nature? You know, we will never know the answer to that. But what we do know is that we're going to continue to see nature doing what nature does.
So Carla, what would you leave the audience with today in our final 2 minutes?
Yeah. Science is hard, okay? And it can be scary for those of us that don't have deep science. I'm not a scientist, I'm an engineer, which is why I like to geek out on the lab design. But science is hard. But science is desperately needed to be able to save the lives of the patients that count on these medicines. So I think that's the first thing. And I would say again to the elected officials, don't be afraid if you don't understand or don't know the science. It's okay, because there are companies out there, there's specialists out there that can help you and provide you information to make the decisions that you need to make. And what I would say is think about the opportunities that your jurisdictions have when you start having a thriving biotech or life science community, and you're not sacrificing safety. Like, you know, there could be some people say, oh, well, you just want those companies because it's something that's, you know, brings money into the city. That's not the case. There's so many opportunities, I think, when you've got that sort of thriving partnership between the cities and companies, what it can do for the schools, what it can do, you know, for the community. And it's really thinking about, okay, are there the measures in place to make sure that the environment stays safe? And that's where we rely on the numerous agencies that are best at doing what they do. And we shouldn't assume that all companies are bad and that all companies aren't going to, you know, have things of that sort. So it's sort of a lot for you guys to think about, but have an open mind and don't be afraid of the science. Don't be afraid of what you might not fully understand. So
thank you.
Yeah.
Let's give Carla a round. So again, I want to thank you for the gift of your time. SAMCEDA, this is a new series for us, Redefining the Narrative. We will be doing these periodically where we will bring information, guest speakers, very topical as to what's happening in our local community. And what we'd like to actually, I want to thank Google for hosting us, The Grove, Javier, the team from The Grove. We now have the opportunity to come together in community with refreshments, bites and drinks, and for all of you to, again, if you haven't met a new person today, that's my challenge to you. And then the call to action is really to go out and share what you heard today and share what SAMCEDA will be bringing forth in the coming weeks with your friends, neighbors, and community members. Thank you so much for coming today.